Hello, everyone. Good afternoon, good evening, and good morning to you, depending upon where you might be joining us from. I am Rob Burgess, and I head up business development for Cyanobiological. And I would like to officially welcome everyone to the next installment in our series called BioTalk Tuesdays, where we hear about cutting edge technology as it applies to both basic as well as translational research. I would like to again officially welcome everyone, and I would like to ask everyone to please type into the chat box your name and where you're from. It's always good to find out where our folks are logging in from around the world. Before we get started with today's lecture, I just want to say we have one housekeeping issue, and that is we would ask everyone, if you have questions, please just simply type those questions into the chat box. And we will withhold all questions until the end of the seminar. And then I'll run through those questions and I'll ask them verbally of our speaker, and we'll try to get to as many questions as possible. So please feel free to ask your questions in the chat box and we'll get to them after the seminar. And so with that, it is my pleasure to welcome and introduce to us our speaker for today. That is Dr. Jifu Li. Dr. Jifu Li is a group leader at the Genelia Research Campus of Howard Hughes Medical Institute. This is based in Northern Virginia in a small town called Ashburn, Virginia. Before joining Genelia just last year, Dr. Li completed his PhD and his doctoral training in neurobiology and immunology, respectively, at Stanford University. Dr. Li's lab at Genelia studies operating principles of cell surface signaling in two sense and response systems, the nervous and the immune systems. To obtain systemic and mechanic insights, he innovates new methods and designs new methods and tools for analyzing cell surface signaling at the systems, cellular, and molecular levels. And the title of Dr. Li's talk today is In Situ Cell Surface Proteomics, Methods, Development, and Applications. Dr. Li, it is a pleasure and honor to have you today, and we will now turn over the screen sharing to you so you may begin your seminar.
Thank you. Hello, everyone. I hope that you can see the screen. Let me know if there is any IT problem. And it's really my great pleasure to be here to share with you our research on in situ cell surface proteomics from our method development to biological applications, particularly in the nervous system. In the evolution of multicellular organisms, including ourselves, single cells assemble into highly organized structures and coordinate their behaviors to achieve biological functions. If we zoom in to tissues, we can see that those cells interact at their cellular interface. Thus, those molecules, such as cell surface molecules, are very important and play crucial roles in mediating cell-cell communication and their behavioral coordination. Here, I'm going to give you two examples illustrating the importance of cell surface signaling in physiological conditions. In the developing nervous system, through decades of studies, we now know that there are many cell surface proteins mediate the attraction or repulsion of axons and dendrites to determine the architecture of neural circuits, and thus lay the foundation for neural computation. In the immune system, many cell surface molecules mediate the recognition or clearance of invading pathogens, as well as the maintenance of immune homeostasis. Of course, the importance and the function of cell surface molecules extend far from like the developing nervous system or the immune system. It can be very important for tumor, for stem cell niche maintenance, and many other biological contexts. And one important feature of cell surface molecule is that they are the major targets of drug development. Because of their accessibility, they are on the surface. They are favorable targets for drug development. And in many cases, cell surface proteins have very high functional precision compared to like intracellular metabolic pathways or transcription factors. They often influence lots of things. Rather, cell surface proteins frequently have very defined certain function for signaling transduction. Thus, targeting them can provide very high precision for manipulation. In addition, in the recent years, there are lots of new innovations, developments of antibodies, BiTEs, CAR-T, and TCR-Ts. Most of them are targeting cell surface molecules. Thus, through comprehensive understanding of the cell surface proteome, together with these new innovations of antibody or the cell-based therapy, we get a chance to actually maybe cure more diseases. And there are many ways actually developed years ago to profile the cell surface proteome in cultured cells. For example, majority of the cell surface proteins are glycosylated. Thus, the glycosylation provides a molecular handle that we can use to pull down cell surface proteins. Another way is to use primary amine reactive chemicals that do not penetrate the plasma membrane. You can use them to label the cell surface proteins. However, these methods are very difficult to be applied to tissues, organs, or not to mention live animals. Dissociation cells from tissue or from organ is basically a disruptive process that if you use pen or other things, you are basically digest the cell surface proteome that you care about. And the culture cell in vitro can frequently change the cell status and their behavior. And on the other side, the cell surface proteome is highly dynamic and environment specific. For example, the cell surface proteome of any given cell is determined by its cell type and its cell state at that time point and its native environment where it is in the tissue. As the gateway for mediating intercellular communication and the physiological function, cell surface proteome is finely tuned and change all the time. Thus, we would like to figure out a way that we can capture the cell surface proteome in intact tissues. To achieve this goal, we collaborated with Alice Ting's lab and Steve Koss' lab, where I was a graduate student in Dr. Li Chunlu's lab at Stanford University. We developed a perioxidase mediated cell surface biotination strategy. In this method, the commonly used enzyme horseradish perioxidase, HRP, is expressed on the surface of a given cell type of your choice. In the presence of low concentration of hydrogen peroxide, HRP converts the substrate biotin phenol into phenoxyl radicals. Those radicals can only diffuse within a short distance and covalently tag nearby proteins with its biotin handle. Thus, the cell surface proteins will be biotilated. And then they can be enriched by streptavidin-based enrichment and analyzed by mass spectrometry. Importantly, although HRP, this enzyme, will traffic through the Golgi or the secrete pathway, this chemical is specially designed to be membrane impermeable, so it stays extracellularly. Thus, the reaction only happens at the extracellular surface of the plasma membrane. And this reaction achieves cell type specificity because we can control where to express HRP even in a complex tissue with many cell types. By using the membrane non-permeable substrate BXSP and the surface target enzyme HRP, it achieves the cell surface spatial specificity. This reaction only takes a few minutes to complete, thus allow high temporal precision. We first applied this method to fruit fly olfactory system. So in the fruit fly olfactory system, I'm going to use this system quite a bit. So I'm going to briefly introduce its anatomy. The olfactory sensory neurons, they reside in the external organs and send their axons to this organ called this brain region called antenna lobe, where they meet their synaptic partners called projection neurons. Those projection neurons conveys olfactory information to higher brain regions. There are about 50 different types of projection neurons and the 50 different types of olfactory receptor neurons. They form this one-to-one matching, like 50 separated channels to faithfully conduct olfactory information transduction, providing a really nice system to study neural circuit development. It's not too complex and not too simple. So it's a really good, nice model widely used in many labs. By expressing HRP in those olfactory projection neurons and run the reaction, we can see that in this reaction group, by neutrality staining, we can see that the dendrites are thoroughly biotinated, while in the control one, there is no neutrality signal. If we zoom in to the cell body region, you can see this ring-like structure here, indicating that only the cell surface is biotinated, but not the intracellular side. And this result is further confirmed by Western blot. After the reaction, we cannot capture actin or Brock pillow, those abundant intracellular proteins, but we can capture cell surface protein and cadherin specifically in the reaction group, further confirming the cell surface specificity of this method. So since it's working, we applied it to 2 different stages of olfactory projection neurons, 36 hours after preparing information, when those neurons are developing, they're building their circuit, and the 5-day adult, when the circuit is mature and they're processing olfactory information. We obtained hundreds of proteins from each stage with partial overlap. Geontology analysis found that no matter at which stage, developing or mature, those proteins belong to plasma membrane cell periphery, confirming the cell surface specificity of this method by the proteomic data. While they have very similar location annotations, the developing and mature olfactory projection neuron surface proteome have very different features regarding their biological functions. The developing one all have like features, functions with neuronal projection development, axon, targeting chemotaxis, whereas the mature one features terms like signaling, localization, transportation, matching really well with the corresponding stage of the olfactory neurons. And this quantitative mass spec strategy we used actually allow us to look at each individual protein and their change from developing to mature. And here in this volcano plot, the x-axis is the mature to developing fold change. Thus, the right part shows the proteins with higher expression at the maturation stage, while the left part shows the proteins with higher expression at the developing stage. You can see that by color code the known synaptic transmission molecules in red and known developed molecules in red, we can see the segregation of those molecules and revealing a global downregulation of neurodeveloped molecules and a global upregulation of synaptic molecules in the developing to mature transition. Although this has been hypothesized by neurobiologists and development biologists for many years, but this is actually the first time that we see real data at the systems level that the cell surface is indeed evolving from the developing to mature stage through at a systems level. And we can also plug in our data to the known protein-protein interaction data set. And you can see here that those proteins segregated from each other by their molecular families or their interacting partners, and they share the change their molecular dynamics. The color code here indicates the mature to developing fold change. Red means upregulation in maturation, and blue means downregulation in maturation. You can see that the chemical synaptic or electrosynapse, all those ion channels, they have the red color, they cluster together. And the neuron developed molecules, they all show blue. So basically, the functional associated molecular complexes, they have coordinate dynamics. The question is whether we can actually get all those information just by RNA sequencing without bothering proteomics. Unfortunately, the answer is no. So we did saturate the very deep sequencing of all those neurons at the same stage as we do proteomics. And here we show the correlation. The x-axis is the mRNA fold change, while the y-axis is the cell surface protein fold change. Although you can see the overall trend, but the correlation is poor, particularly for those functionally important molecules that we know, those blue and red dots here, you can see that many of them are far away from the regulation line, and even some of them show opposite trends in the RNA and the protein change. And this is not surprising to us because from mRNA to protein on the cell surface, there are lots of translation regulation, post-translation trafficking regulation, protein turnover regulation. So you need to look at the protein's name directly to get the information there. And now we are going to zoom into the biological part, whether we can get useful information from the proteomic profiling. So here I list the top 100 cell surface proteins enriched at the developing stage. To our surprise, and actually a happy surprise, that in the past 2 decades, many labs use the olfactory system to search for cell surface proteins controlling neurodevelopment. And in this single chart, in this single experiment, we captured almost all of them. And here are the citations of all the previous discoveries. This really inspired us that whether we can use this dataset to efficiently find new wiring molecules. So we went back to the volcano plot. You can see that at the left corner, the blue dots are enriched there. And there are lots of like the gray dots, those unstudied molecules there. Whether those molecules also participate in neuronal wiring or not. So we tested them. Previously, the lab devised method to do screen, in vivo screen, and we conducted a large-scale screen covering almost 300 proteins and identified 15 new ones. And to our surprise, using our new methods, the proteomic guided screen instead of the molecular family guided ones, we achieved a very high hit rate, 20 out of 20. And here I'm listing all the 20 newly identified wiring molecules. You can see that many of them have human orthologs. Importantly, lots of them, majority of them highlighted in yellow here, came from molecular families that were never discovered in neurodevelopment context, including chitinase, transporters, scavenger receptors, and many others. This maybe partially answer why it is so difficult for us to find the new wiring molecules in our previous way by molecular family guided candidate screen. Those molecules may hide in those molecular families that we out of our imagination. And here I'm giving you one specific example. Here we found that LRP1 controls dendrite targeting in fly brains. And LRP1 is actually a famous molecule for another reason, is also the ApoE receptor. So the one that is highly associated with neurodegeneration and cardiovascular diseases. And you can see that the fly LRP1 is highly conserved compared with the human LRP1. And we found that by knockout LRP1 in the fly olfactory neurons, you can see that in this neuronal type, the dendrite spill over to the nearby regions. While in the other ones, neuronal type, it targets a very far away area, show very distinct but highly specific phenotype in distinct neuronal types. And this is the first report of LRP1's function at this very early stage in neurodevelopment. And so far, we still don't know how it does this job. Inspired by the success of the previous study, then we actually want to use this method to ask a very different question, to try to map out the transcription factor to cell surface executor to physiological function access. In the biological system, transcription factors are often the master regulators of cell fate and behaviors. However, they do not mediate cell-cell interactions directly because they stay in the nucleus rather than at the cellular interface. And for example, in the fly olfactory system, half of the projection neurons express this master regulator called ACJ6, which is a podomain transcription factor. It specifies half of the PN lineage and controls the targeting of all those PNs, but they do not mediate this process by themselves because they stay in the nucleus, they are not on the surface. Thus, we believe that the molecules actually direct mediating a neuronal connection is on the surface and controlled by ACJ6. Considering that it's a transcription factor, we originally thought that we can use RNA sequencing to directly map out the downstream molecules. So we did RNA sequencing to compare wild type and ACG6 mutant PNs. And among the 5 top families of proteins or genes regulated, we found that one is protein processing in ER, and the other one is endocytosis. Basically, ACG6 regulates many genes on the way that the protein is going out on the membrane and also on its way to getting back in the recycle pathway. Thus, ACG6 can control protein delivery to the cell surface through these pathways, and it controls the cell surface proteome through at least 2 indirect ways. One is the direct regulation of their transcription in nucleus, and the other way is through the indirect pathways like the ER protein process and endocytosis. Thus, RNA sequencing would not be able to reveal the actual cell surface change in ACG6 mutant. We have to look at the proteins directly. Thus, we profiled the cell surface proteome of wild type and ACG6 mutant and found that indeed there are many proteins regulated by ACG6 here. And interestingly, we found one molecule called Piezo. What is Piezo? Piezo is a mechanotransduction ion channel discovered by Adam Patapoutian and his team many years ago. And since its discovery, it has been a popular hot molecule in the field because it controls many processes in the somatosensation, regulation, and other pathogenesis process. Why would such a central nervous system neuron, which is not mechanosensitive, express piezo at the developing stage and controlled by this ACG6 transfer factor? Would it be possible that piezo actually controls dendritic targeting? That would be very interesting. So we tested this. And in these neurons, in wild type flies, they do not target to these brain regions. And in ACG6 mutant, they mistarget. And when we introduced the piezo knockout, they mistargeting in a very similar way to ACG6. Importantly, when we supply back piezo, we find that it fully rescues the piezo mutant phenotype as well as the ACG6 mutant phenotype, indicating 2 things. Piezo is indeed controlling dendritic targeting, and second, it's the downstream of ACG6. How would the piezo do this? It's a mechanosensitive ion channel. Would it be possible that the piezo does this job through mechanosensation? To test this, we generated a piezo mutant that is insensitive to mechanical stimuli. And with the help from Dan Olin from Swiss Mercy's lab, we verified this mutant and see that by hook the wild type piezo transfected cells, we can see this current, and this piezo mutant is completely silent. And when we supply this mutant piezo back, we found that it fully rescues the piezo mutant phenotype as well as the ACG6 mutant phenotype to a similar extent to the wild type piezo. There is no difference, indicating that piezo does this independently of being a mechanosensitive ion channel. And so far, we still don't know how it does this job. It's a very interesting question. Beyond piezo, we also found that the dopamine receptors, DOP1R1 and DOP2R, are expressed at developing PN surface and also regulated by ACG6. And through RNA knocking down, we found that both of them give very similar phenotype to each other that cause mistargeting or loss of targeting in dendritic targeting. And extending from piezo and dopamine receptors, this proteomic profiling allows us to study many molecules in many neuronal types, and we focused on those ones downregulated in ACG6 mutant and examined their phenotype in this all 11 PN types here. And here we're not going to, because it's so many molecules and so many neuronal types, I'm not going to show you the phenotype one by one. Rather, we're going to show a color code map that the control is showing this white color. And we found that ACG6 mutant causes mistargeting all 11 PN types, and we're going to show a red color. And then we supply back those cell surface molecules one by one to see whether they can rescue the ACG6 mutant phenotype. If it's a complete rescue, show it in the white color. If it's no rescue, it's red, similar to the ACG6 mutant itself. And if it's a partial rescue, it's pink color. And we found that by supplying back one isoform of ACG6 itself, it rescues most but not all phenotypes. Using the piezo as an example again, When we supplied back piezo, it specifically rescued these 3 PN types partially, but not the others. As shown in each row here, each cell surface molecule partially but specifically rescues the phenotype in a few PN types. While showing in each column here, each PN type frequently requires a few distinct wiring molecules for its correct targeting. This mosaic matrix immediately reminded us of this decade-long hypothesis of neuronal wiring called a combinatorial coding hypothesis. Think about our genome. It only has about like 20,000 genes there, not to mention that majority of them are not cell surface molecules. However, they need to specify neuronal connections in our brain with like trillions of neuronal connections. If they use a one-to-one system, one gene, one connection, they will soon run out of molecules. We would never be able to wire our brain. Thus, there is a hypothesis that those molecules can form combinatorial codes. Thus, their coding capacity can be extremely expanded in this way, and then they can specify more neuronal connections. This data here immediately remind of this combinatorial code and provide us with the opportunity to test this hypothesis by experiment. Thus, we supplied back those molecules paired in pairs and observed that indeed by combinatorial expression of pairs of the cell surface molecules, observed better phenotypic rescues, as shown here by more white and light colored squares here, providing a clear evidence that there is indeed a combinatorial code. And by zooming in to this matrix, we can actually observe the interaction modes of those molecules. For example, the OTK and the piezo, both of them provide partial rescue of the ACG6 mutant across the phenotype. And we will add them together. They provide almost a full rescue of the phenotype, forming an additive interaction. And for these two molecules called OTK and DOP1R1, you can see that by themselves, they don't provide statistically significant rescues. However, by putting them together, they provide significant rescues, indicating that they may require each other for their function. So they have a synergistic interaction. And you cannot just randomly add molecules together, or you need to have the right pair, because as shown here in this DG and NEP3 case, NEP3 actually can rescue this phenotype significantly, but DG actually makes this phenotype even worse. By combining them together, it can actually make the scenario worse than NEP3 itself. So you do need to have the correct combination. And taking everything together, we actually mapped out the transcription factor to cell surface executor to neuronal wiring, this axis, by using ACG6 as an example. This single transcription factor leverages many cell surface molecules, actually specifically through their combinatorial code to specify the wiring of many neuronal types here. And this is actually the summary of the real data. We hope that this approach we take can inspire the study of many other biological questions. For example, how do a core transcription factor specifies the fate and interaction of stem cell through its cell surface molecules, and many other questions. And you might have been wondering, I demonstrated all those things in slides, whether it works in mammalian system or not. The answer is yes. So through a collaboration with another graduate student, Andrew Schuster, we developed this transgenic mouse model by knocking the PCAG-driven HRP into the universal ROSA26 locus. And this mouse achieved the cell surface expression of HRP and also cell type specificity gated by Curie flip or the combination of them. And this mouse model is available at JAX for everyone's use. And we first tested it in multiple tissues and cell types and found that by neutrality staining in green, cell surface biotination can happen in kidney, heart, liver, fat, intestine, opening the opportunity to lots of scientists in these different systems to use this mouse model. And we first tested in our favorite system, the cerebellar Purkinje cells. If we omit HRP or the hydrogen peroxide, you can see that there's no green staining of neutrality, there's no biotination. And when we run this reaction in the Purkinje cell, you can see thorough biotination of the whole molecular layer where the Purkinje cell dendrites are. And if we zoom in to the cell body region, again, you can see this ring-like structure of this Purkinje cell body, that there's no intracellular labeling, just the cell surface. And we obtained hundreds of proteins from the developing and mature Purkinje cells with partial overlapping. And similar as the fly case, this data confirmed that those proteins are localized to the plasma membrane and the cell periphery. And importantly, we found that those proteins, no matter at which stage they're developing or mature, we can enrich transmembrane proteins, GPA-anchored proteins, and secretory proteins, basically all three major families of cell surface proteins there. Similar to the fly case that we enriched this in the surface proteins. However, very interestingly, that if you still remember the fly data, most of the neurodevelopment or adhesion molecules go down after maturation, and the synaptic molecules go up. We still observed the similar trend here in the mouse case of Purkinje cell, that the developing molecules are enriched at the early stage, while the ion channels and others enriched at the later stage. However, biological adhesion molecules stay always very high throughout the process. And one possibility is that if we look at this classic drawing by Cajal of the Purkinje cell, those neurons have very dedicated dendrites arbors, and maintaining these arbors may require actually biological adhesion throughout the lifetime. And the question is whether the adhesion molecules are the same or different at the developing and mature stage. So we zoom into our data again by the volcano plot. And here we highlight the top upregulated proteins in maturation and top downregulated ones. You can see that the synaptic molecules, most of them are upregulated in maturation as expected. Regarding the cell adhesion and guidance molecules, a group of them got downregulated in maturation, while the other group got upregulated in maturation, indicating that there is a switch of cell adhesion and guidance molecules in the developing to mature process. Although we don't have genetic evidence for this, but we can anticipate that these ones we may participate in the developing stage, and these may be required for plasticity or maintenance. And this data inspired us that whether we can actually find new molecules controlling Purkinje cell development, and we focused on those uncharacterized, unstudied molecules. And actually, we found one called RH4, which gave very strong phenotype when we knocked them out. In wild-type animals, Purkinje cells grow this very beautiful dendritic arbor shown here. And when we knock out RH4 by CRISPR, you can see that the dendrite is much shorter and it has less complex dendritic arbors. The interesting thing showed up when we tried to overexpress this molecule in Purkinje cells, RH4, and we found that overexpression of RH4 show very similar phenotype as the knockout. It has much shorter dendrites and less complex dendrites. Seems that the existence of RH4 is not enough, is not sufficient. Dendritic arborization is very sensitive to RH4 expression level. If you don't have it, it does not grow well. If you have too much of it, it's bad. Then we tested whether the intracellular domain, since it's a transmembrane protein, whether intracellular domain of RH4 is required for this phenotype or not. And we removed the intracellular domain of RH4 and overexpress it, and the phenotype is gone. The dendritic arbor goes back to the normal status, and indicating that the intracellular domain is indeed required for RH4's function. But when we do the staining of RH4 by HA, a tag, we found something really interesting, that in the wild-type RH4 overexpression, you see those proteins form puncta intracellularly, while the intracellular domain, the ICD knockout RH4 expression, the proteins are definitely on the surface, including in the dendritic spines. Seemed that there, besides the signaling transduction, there is something in the intracellular domain, in the ICD domain, control the protein targeting. So we zoom into the intracellular domain and actually found a 6 amino acid endocytic motif in the intracellular domain. that may control the protein targeting. So we tested this. And by only remove the 6 endocytic motif to alanines, we found that now when we overexpress it, proteins still go to the cell surface and with actually showing much stronger dendritic phenotype than the wild type overexpression. So now we observed that there are at least two parts in the ICD domain. One is that there is the 6 amino acid endocytic motif controlling protein targeting, and there is an unknown signaling transduction motif there required for RH4 signaling. Taking everything together, what we observed is that RH4 get endocytosed into endosomes, which regulates the level of RH4 on the cell surface. When we overexpress RH4, it disrupts the dendritic morphogenesis. However, because we still have this regulation mechanism, this level on the surface is controlled. When we specifically remove the endocytic motif, all the proteins goes to the surface without actually endocytosis downregulation. So the dendritic morphogenesis is severely disrupted. However, when we remove the whole IC domain, although all the proteins goes to the surface, but the signaling transduction motif, also which remains to be discovered, is also removed. So you don't get any dendritic morphogenesis phenotype. Looks similar to the Y type. And taking everything together, we have shown you that we developed a method for in situ cell surface proteomics to do it in fruit flies and mouse, and through the cell surface proteomic profiling, we can identify many interesting molecules controlling neuronal development. After the release of our method, actually we received a request from many users saying that it's so difficult to analyze the data. We don't have experience with systems biology. We don't know how to analyze proteomic data. Thus inspired us to build an integrated and user-centric web server for the cell surface proteomics and actually overall spatial results of proteomics data analysis. So recently we released the website called Piezo, and it's also available through the PyPy on the Python website. And it's a really simple web server. And what you need to do is to just choose the cellular compartment, for example, cell surface, supply the data file, and then tell us a little bit about your experimental design. Then just one simple click here, submit. It will do a whole pipeline for you, including the quality check by correlation analysis, whether the data, the biological replicates correlate with each other. Then it use curated references to check whether you actually enrich the proteins in the designated cellular compartment and perform cutoff analysis. Then it give you the post-cutoff proteome and retrieval the protein information from UniProt database to let you know what are those proteins, their lengths, and basic information. And it further sends your proteome to geontology analysis and let you know whether your protein is indeed localized to the cellular compartment you wish to profile. And what are those proteins do there based on the geontology analysis, and what are the enriched molecular pathways there. So bring everything together, we started from developing a wet lab, actually chemical biology method to profile cell surface proteome in intact tissues in animals, and then extend from the wet lab work to the dry lab part. Now we provide a full pipeline from wet to dry toolkit for in situ cell surface proteomic analysis. And with this, I would like to thank all my current lab members, my collaborators at Genelia, and particularly lots of work were done when I was a graduate student postdoc at Stanford, my PhD advisor, Li Chunlo, and my collaborators in the lab, Ricardo, Chichi, Andrew Tran, and Hongji, and my long-term collaborator, Alice Ting and Steve Carr and their teams for the chemical biology tool development and mass spec collaboration, as well as with the Merthy's lab at the Volume Institute for collaboration on electrophysiology characterization of Piezo. And with this, I'd like to take any questions. Thank you.
Thank you, Dr. Lee. That was an excellent seminar. We appreciate it. Very cutting edge technology for identifying those cell surface receptors in a very comprehensive fashion. Our first question comes from Gopal Pathak, if I pronounce your name correctly. And Gopal actually has two questions. And the first one is, how can he utilize the technology that you've developed to identify differentially expressed proteins on the surfaces of patient-derived tumor tissue compared to matching normal tissue? So is it possible to use the technology on tumor tissues as well?
Yes, so this is a good question. So if it's a mouse model, it will be very straightforward and easy because you can always transgenically express it. For human samples, that's a very interesting question. It's very important too, that if you can dissociate those cells and culture them, then you may be able to actually also use the HRP-based labeling. But if you want to directly do it, I would say that either use the primary amine-based cell surface labeling method or some other chemical labeling, although you lose the cell type specificity there, but that's probably the only way. Yes.
Great. Thanks for that answer. And then Gopal's second question is about the sensitivity of the system. And specifically, he asks, what is the sensitivity of detecting or identifying an unknown protein on a cell surface? And what Gopal means by this is he's referring to the ability to detect like very low levels of proteins. Do you know the lower limit of detection?
That's a very good question. So we actually don't know like how low we can go, but with the current improvement of mass spec itself, actually you get a very deep coverage. Frequently, through the cell surface proteomic profiling, we can detect thousands of like 3,000, 4,000 proteins there, and some like 800-ish are likely cell surface proteins. So even for the lower abundance one, with the improvement of mass spec, I think the detection chance is very high. Yeah.
Great. Thanks for that. And the next question is from Kabita Sharma. This is kind of a long question. I'll see if I can ask it of you properly. So how mechanosensitive piezos and other cell surface molecules, how do they respond to change in a cell membrane's potential? Like, for example, the change in the potential near the synaptic cleft or the synaptonemal complex that might occur like during an injury or an infection of those cells. So how would those cell surface molecules respond to that?
That's a very good question. We never tested it, but we believe this method can be a good one to actually try to profile that. Yeah, it's a very interesting biological question.
Yeah, sounds good. And there was another question about the use of human surgical samples, but I believe you answered that very nicely about the tumor tissues there. So still waiting. We've got a few minutes left. If anybody would like to ask any other questions. I have to say, Dr. Lee, I'm a mouse geneticist by training, so I was very happy to see your use of the transgenic mice, the Cree-specific, tissue-specific flip FRT sites, the Rosa 26 locus always brings back great memories for myself when I was studying that at MD Anderson in Houston and at Lexicon Genetics a few years after that. So kudos. It's great to see the mouse model still being actively utilized to study gene function. And with that, let's see, we have some other questions here. Just 1 second. Brenda Ben Su is asking, does the labeling affect the immune function on the immune cells?
So basically for the labeling, it completes in like 10 minutes, including everything. So after that, you either add PFA to do histology or you freeze them down. So it doesn't matter whether it changed the function, but because you add biotination onto the proteins, I assume that it will do something there.
Right. Murugesan Velayuthan, please forgive me if I didn't pronounce your name correctly, is asking the question, can we detect the phenoxy radical using EPR method?
I'm not sure what EPR is, so I cannot answer this question. Yeah.
Okay.
Sorry.
So Muru, if you can clarify what EPR is, we can get back to your question. Gorak Giri is asking, what are the ways to detect differentially located proteins in cell organelles or cytoplasmic ones rather than just cell surface proteins? Is there a way to do that?
Yeah, definitely. I mean, that's widely used by using those methods like BioID, Apex, or TurboID. You can, you can simply do that. It's very robust. Yeah.
Great, great. And Muru got back to us. Muru says EPR is electron paramagnetic resonance spectroscopy.
That's beyond my knowledge, so I'm not sure whether we can detect through that. Yeah.
All right. Appreciate your honesty there. Few more minutes left. If anybody has any other questions while we're waiting, let me ask you, Dr. Lee, is there an opportunity to translate your developed technology into like a startup company or a business any nature? Is it really more applied towards basic research?
Yeah, actually, so for this tool, we originally designed it for basic research, but now actually it's adopted by quite a few companies that I'm aware of that they are using this to study neurodegeneration diseases, trying to find biomarkers, trying to find therapeutic targets. So indeed, it has been used in the industrial field to actually, you can say that it's actually very good for biomarker discovery.
That's great. So you've actually licensed your technology out to some companies then?
Yes. So it has been licensed. Yeah.
Great, great. All right. So I think we have one more question here, then we'll wrap it up. Moni Nader is asking, it's first of all, Moni says it was an excellent talk. Thank you for that, Moni. Is the intensity of the labeling cell specific? For example, would the abundance of the glycocalyx on the surface membrane affect labeling or sensitivity?
That's a good question. We never specifically tested, but because radical reaction is so strong and robust, I think based on our observation, no matter in culture cell, in fly brain, in mouse brain, and in many other organs, seems that it's universally robust throughout it. Well, as long as you can get HRP and the probe delivered. Yeah.
Great. Moni says, thank you. So with that, I think we'll wrap things up just a few minutes early, if that's okay. First of all, I'd like to thank all of the attendees. We had a great turnout today, people from all over the world. I just briefly want to say where everybody called in from. Cayman Islands, UAE, Germany, India, all over the US. I won't name all of the states there. Nepal. In fact, I was just in Nepal last month, so that's very cool. Algeria, Uganda, Morocco, Indonesia, Colombia, Canada, and Brazil. So really a worldwide attendance there, and especially those of you, it's middle of the night or early morning, we appreciate you attending. Dr. Lee, certainly want to thank you for an excellent seminar. We appreciate it, very cutting edge technology. So congrats to you on all your personal success and your team and all their success as well. And I'm sure we'll be hearing about more very exciting research from you and your team in the future.
Yeah, thank you so much, and I really appreciate the opportunity, and I hope that these tools can be helpful for your research things. Yeah,
fantastic. Great talk today. And one more thank you. It goes out to my colleague, Max Bleckman at Sinobiological. Max organized and executed this online webinar. So thank you, Max, as well. And I will wish everybody good evening, good morning, or good afternoon. Bye-bye.